All right, let's continue. Last time we ended on this slide 23 here of 47 in Unit 7A. So this is Lecture 7A, Part 2. I'm going to talk to you about the motor unit. This is the functional unit within muscles. When we talk about functional units, it's typically the smallest part of the organ system that carries out all of the functions characteristic of that organ system or physiological system. In this case, for muscle, it is the motor unit. It's a combination of the motor neuron and the skeletal muscle fibers that are innervated by that motor neuron. Now, the number of actual muscle fibers within a single motor unit, in other words, the number of muscle fibers that could be innervated by a single motor neuron, can vary widely from as few as 4 to up to several hundred. Those muscles that control very fine movements like the tongue, the lips, the fingers, the eyes have small motor units, whereas muscles that are involved in more cruder kind of contractions like your thigh muscle in your legs have many more muscle fibers associated with each motor unit. Now the muscle fibers innervated by a single motor neuron, in other words, the single motor unit, the muscle fibers are not all bunched together in one area, they're spread throughout the muscle. So what you get with activation of a motor unit, one motor unit, is a weak contraction overall of the whole muscle. And as more get activated, then the contraction strengthens, but it's spread out throughout that entire muscle. Force of contraction can be affected by the number of muscle fibers contracting. So obviously the more motor units that are activated, the stronger the contraction. The relative size of the muscle, the bulkier the muscle, the greater the contraction strength or force generated. But another important aspect that isn't quite as intuitive is the fact that depending upon the length of the muscle at the time that the contraction starts, you can get more or less strength in that contraction. If a muscle is stretched out to its full length, you won't get a strong contraction. If it's shortened a lot, you won't. But if it's at 80 to 100% of the normal resting length, that's the range in which you get the strongest contraction, the most force generated. Okay, now the next few slides describe experimental aspects or values that can be obtained when you isolate muscle from a living organism. And we actually used to do this in the lab until they decided to save the frogs of the world and stopped us from being able to do it. But we used to take a muscle out of the leg of a frog that had just been killed and suspend it suspended on so that a force transducer could measure the amount of force generated. Now you can do that two ways. You can do it with just the muscle and then stimulate it with a voltage stimulus directly onto the muscle, or you can dissect it away, keeping the nerve are still attached to the muscle and then stimulate the nerve. Muscle tissue actually is quite tolerant of hypoxia, low oxygen. Um, Amazingly tolerant, actually. It can go a long time in a fairly hypoxic state, partly because of mostly I would say, but mostly because of the myoglobin in it that contains oxygen. So you can excise it out of an animal and suspend it from a hook that's attached to a force transducer, and the opposite end you can put different weights and see how much contraction force, or just suspend it between a fixed point, a hook, and a force transducer, so it'll measure clearly how much pull there is. The smallest stimulus possible, a single brief stimulus that generates the smallest contraction, that contraction is called a muscle twitch. So you use the smallest stimulus possible to get a contraction in the muscle, and that is the muscle twitch. If you go below that stimulus, you haven't hit the threshold required, and there's no contraction whatsoever. But as soon as you get to a strength of a stimulus that'll generate a contraction, that contraction is considered a muscle twitch. So here we have a characteristic graph showing the kind of force or tension that can be generated with a single stimulus that triggers this kind of muscle twitch. Now, by convention, the length of the stimulus arrow, the arrow indicates the stimulus, the time at which the stimulus is given, and the length of it indicates the strength of that stimulus. So if you see a short arrow and then a longer arrow, that would indicate, like a longer arrow, not here, but if you gave another stimulus again and the arrow this time was longer, then that would indicate a larger stimulus. So the characteristics of this muscle twitch are as follows. There are three periods. The first is the latent period, which is the period between the time that the stimulus is delivered and the first appearance of any force or tension. Then the period of contraction, in which there's an increase in that force going all the way up. Let's do this here, going up to this point here. So that's the period of contraction. And then the period during which the force or tension generated is reduced as the muscle relaxes, and then it goes down to zero. Note this is in milliseconds here. So that's the latent period, the first few milliseconds when nothing is happening, and then the period of contraction. And during this period of contraction, there are cross bridges actively formed. What this is referring to are the temporary connections being formed between myosin heads and the myosin binding site on the actin of the thin filaments. So thick filaments are the myosin heads and the thick filaments are attaching, sliding, ratcheting the thin along the thin filament, and then detaching, then attaching. So there's a shortening that way. Those are the cross bridges. The muscle is shortening. During the last period, remember that it is calcium released from the sarcoplasmic reticulum that triggers that myosin-actin interaction that allows for shortening. So during the period of relaxation, calcium is not being triggered to be released anymore, and in fact is being reabsorbed into the back into the sarcoplasmic reticulum, and muscle tension drops back down to zero. That's the period of relaxation. Okay. You can look at muscle responses in this kind of a setup and see that there are certain key characteristics. You can get graded muscle responses, or in other words, varying degrees of contraction. And that is, in fact, what's required for proper control of skeletal muscle movement. You know, you can lift a heavier object if you increase the amount of muscle contraction. And that's what you do when you lift something heavy. If you don't generate enough force, and you're trying to lift it, then you'll stimulate more and contract the muscle, allowing the muscle to contract with stronger force and be able to lift something heavy. These responses are graded by change in frequency of stimulation and change in strength of the stimulus. Now we can look at this kind of responses in muscle in the same kind of setup using an isolated muscle, and look at these kind of tension versus time graphs or data generated with different types of stimulus, either changing in strength or changing in the frequency of the stimulus. Here what you see is a single small stimulus being given with the latent period and the period of contraction followed by the period of relaxation, a single twitch. Now, if a stimulus is given before the muscle completely relaxes, then you get what is called a wave summation. So before it completely, as it's coming down into this period of relaxation, another stimulus of equal size is given, then you'll get a larger increase in force. This kind of characteristic response is called the wave summation. And wave summation is, in fact, although there's another characteristic feature of response of muscle that's somewhat similar, but don't get it confused. Wave summation is in fact due to the fact that not all of the calcium is being pumped back. So with the second stimulus, you have increased amounts of calcium available, and it goes higher and then higher. If you start increasing the frequency, you start getting what's called tetanus. The muscle starts to quiver, and you're constantly getting this force generated with small, increasing strength as you give each stimulus. It's a little bit subtle, the difference between these two, I'll admit, but there is a difference in that this is talking about the wave summation really refers to this kind of increasing force generated, whereas tetanus is unfused, or incomplete tetanus is trying to describe what's happening just before you hit a frequency over here, right? That's incomplete tetanus on the left-hand side here, and on the right is complete or fused tetanus. If you keep increasing the frequency of the stimuli, as shown by these red arrows on the bottom, eventually the muscle stops quivering, contracting and relaxing, and is just constantly tense. So there is some subtleness between these two, but this is what's called here wave summation. This is unfused or incomplete tetanus, and this is complete tetanus, where it's completely tense. It is true that, that there is an infectious disease caused by a bacteria, which causes the disease tetanus. And it's called, the disease is called tetanus because in that disease you get disruption, or it affects the neuromuscular junction so that there's continuous stimulus, and you get this kind of constant cramping of muscles. Okay. Now, as I told you, if you stimulate a muscle and you don't give enough voltage, then you won't get any contraction. If it's a subthreshold stimulus, you won't get any contraction. And then as you increase the stimulus, electrical stimulus, you'll hit the threshold. And that's the first stimulus strength at which you can see observable muscle contraction. Once you go beyond that, then the muscle will contract even more vigorously because you get more motor units recruited into that contraction. Force of contraction, in fact, we control the force of contraction of our muscles by carefully controlling motor units, and you get, in the end, you get a summation of motor unit activity. So what we do is we recruit more and more motor units, and therefore more and more muscle fibers, as we try to increase the strength of muscle contraction or generation of tension. And again, the example of that in real life would be trying to lift a weight but not generating enough force in your muscles so you can't lift it off the table, and then consciously recruiting more motor units and generating more force and lifting it up. That's called recruitment. And this is these are graphs that show that kind of thing. These are the size of stimuli, the length of the arrow of these columns, rather, in this case, in this graph, shows the strength of the stimulus. If it's sub-threshold, you're not going to get any force or tension. There's no contraction. As you can see, these are here sub-threshold stimuli, and therefore there's, if when measuring tension, these two graphs are being measured simultaneously. So there's the stimulus being delivered and the size of the stimulus, and here's the force being generated, the size of it. Once you hit threshold, you get a twitch. So you're getting a single motor unit activated. Then with a stronger stimulus, another motor unit gets activated, and it increases in size and more. If you gave the maximal stimulus, increasing it further wouldn't change anything. And again, just to remind you, this is a motor unit, a motor neuron here, axon. It innervates these two muscle fibers, and you'll stimulate that, and you'll get a contraction. Some motor neurons will stimulate more. The motor unit is innervates more muscle fibers. So with stronger stimulus, you recruit those, and you get a stronger generation of force. Okay. If you get a single action potential sent out along the motor neuron, and it arrives at the muscle fiber and triggers a contraction, then you'll get a twitch. If the twitches are occurring in rapid succession, you can get increase in the total strength. It has to be close to each other, and that's sort of like a summation. Tension is generated and increases and becomes more sustained as the signals come in faster. In fact, as I said before, what you're getting is a higher rate of stimulation, and associated with that, more calcium being released from the sarcoplasmic reticulum, and in between there's just not enough time to remove all of that calcium from the sarcoplasm back into the sarcoplasmic reticulum. Sarcoplasm, I remind you, is the cytoplasm of the skeletal muscle fibers. So it can't get pumped back in, and you start getting, first you get wave summation, then you start getting tetanus. Eventually you'll get tetanus, and maximum tension is generated. Well, you can't generate maximal constant contraction of skeletal muscle. You can't get tetanic contraction unless you have ATP, and a lot of ATP. And eventually that supply of ATP will be exhausted, and muscle fatigue will start setting in. Okay? Everything depends on the availability of ATP. So here we see stimulus. Um, you get stimulus and you get generation of force, and it goes down, and then another stimulus, and I don't see an increased wave summation. But here, because the stimulus came first here and then soon after there, before it could completely relax, then you get this wave summation. And here you get an example of rapid, even more rapid stimuli. So you're getting wave summation going here, and the effect actually is generating an incomplete tetanus until eventually it's the frequency of stimulation is increased fast enough so that you get complete tetanus here. So you have to keep in mind that muscle contraction or tension can be generated in a number of ways, actually. Now, when skeletal muscle is contracted, often different degrees of strength or different levels of strength are required. And that can be generated in a number of different ways. It all, in the end, depends on the number of muscle fibers that are recruited in the muscle. But one way of making a stronger contraction or generating more tension or force is by increasing the number of motor neurons that are firing at any time. And that would increase by increasing the number of motor neurons, then you're causing spatial summation. So they're all acting within the particular muscle, and they're acting at the same time, but you're recruiting more motor neurons and therefore more motor units. Another way is to increase the rate of firing of the motor neurons, and that contributes to temporal summation. So you get more of them firing very closely. You will generate more energy, more tension. There are many muscles in the body, and we talked about this actually with the heart, that there's a we talked about this with smooth muscle, that you often have a certain degree of smooth muscle contraction, for example, around blood vessels. So vascular smooth muscle often has a background amount of contraction occurring so that it can undergo either vasoconstriction or vasodilation by relaxing, by contracting more to generate vasoconstriction, or relaxing to generate vasodilation. And neither of those is possible if the muscle was fully contracted all the time or fully relaxed all the time. So a lot of muscle tissue in the body is undergoing a low level of tension at all times, and that's called muscle tone or tonus. It's due to a small number of motor units in the skeletal muscle. It's due to a small number of motor units being activated, regularly activated at different times. Now, there's another effect that can be observed looking at isolated muscle and force transduction in a muscle that gets stimulated with voltage, and it is called a staircase effect, or TREEP. This is an increase in contraction in response to multiple stimuli, and it can be seen and is demonstrated when the stimulus is given immediately at the end of the period of relaxation. So the muscle gets completely relaxed, and then the stimulus is given. And what you get is an increase in the strength of contraction. There's more tension or force generated, and it is because of two reasons. One is there's an increased availability of calcium in the sarcoplasm, because again, not all of the calcium has had enough time, even though the muscle was completely relaxed, not all of the calcium had enough time to be pumped back into the sarcoplasmic reticulum. The other is that many enzyme systems, including muscle enzyme systems that are involved in the generation of ATP, are more efficient when they are warmer at a higher temperature. And contraction of muscle will cause an increase in the temperature of the tissue. The conversion of chemical energy in the ATP to energy involved in the contraction, the contractile force, is not 100% efficient. A lot of that energy is lost. In fact, a very large proportion of the energy is lost as heat. So as muscles contract, they generate heat, but there is a benefit to that in that the enzyme activity improves. So the enzymes involved in formation, the pathways involved in formation of ATP in the muscle, and we're talking mostly aerobic respiration, things like glycolysis, pyruvate oxidation, Krebs cycle, electron transport chain, all of them are improved in efficiency. The enzymes involved in them are improved in efficiency as they warm up. You no doubt remember seeing a graph that looked like this, where the horizontal axis is temperature and the vertical axis is enzyme activity. Let's call it enzyme activity. These are typically measured by looking at product production, the product of a particular enzyme. And typically they have a characteristic profile. Let's do it with a pen. They have a characteristic profile like that, where there is an optimum temperature at which the enzyme is able to produce the most amount of product. So as it heats up and you move, say, from here to here, as the muscle warms up, then you increase in efficiency. And that is a large part of the generation of that, or the large part of the cause of the staircase effect. Improved performance occurs because of these two mechanisms, or these two mechanisms operating in muscle that has been warmed up by continual stimulus, and can be demonstrated by stimulating the muscle immediately at the end of the period of relaxation. And it looks as shown. Oh, I don't have it. Oh, there it is. I missed that previous slide. I skipped through the previous slide and I don't believe I can go back. So I'll have to look later what was on it. In any case, here, this is what you see with the staircase effect, or TREEP. The stimulus is given here initially, and you get this kind of force generator tension. The same size stimulus, no different, is given right at the end of the period of relaxation, and it generates a higher tension due to the two characteristic features. One is not enough time to pump all the calcium back into the sarcoplasmic reticulum, so availability of calcium rises. And the second is that the tissue is warming up, and therefore the enzymes are acting more efficiently. And this really is the reason that athletes should in fact warm up prior to a competitive event, a long-term competitive event. It's not just this idea of stretching, but actually warming up the muscles so they work more efficiently. Okay, let's talk about production of energy. How do muscles get ATP? Where do they get it from? ATP is the source of the energy that muscles need to to contract and generate tension or force. There is some, not a lot, but there is some ATP present in the muscle tissue itself. It's not a huge reservoir, but it is there. But when muscles contract, they quickly, within just a few seconds, 5-6 seconds, consume whatever's there. There is another source of essentially almost immediate source of ATP, and that comes from creatine phosphate. Creatine phosphate is phosphorylated. It has a phosphate group, and the bond to the phosphate contains quite a bit of energy. When you first start contracting muscle, immediately the ATP here gets used up, and then the muscle will start using creatine phosphate as a source of production of ATP. It's a very quick way of producing ATP because the phosphate attached to the creatine is donated to ADP to form ATP. ADP, of course, is adenine diphosphate. ATP is adenine triphosphate. So the muscle can form ATP from ADP, and this is a process, the general term for this kind of process is here, that's occurring here, is this kind of generation of ATP is called substrate level phosphorylation. Substrate, takes time to write these. Substrate level phosphorylation. So this is not a method that lasts for very long. The amount of ATP that can be generated from creatine phosphate stores, it's important, but it's valuable, but it only lasts about 15 minutes, and then the creatine phosphate stores are exhausted. By far the largest amount of energy produced in muscle, by far the largest source of energy, ATP, produced in muscle occurs or is derived from the process of aerobic respiration. So when we're talking about aerobic respiration, we're talking about the use of carbohydrates, the energy in carbohydrates, and also sometimes from fatty acids, but mostly from carbohydrates to generate ATP. And this is a process, it's called aerobic because it involves oxygen. And I remind you that oxygen, that's why we breathe oxygen, so that we can carry out, your cells can carry out aerobic respiration. Not just muscle, but all of your cells need ATP, and they can generate it by aerobic respiration. So carbohydrates are broken down, can also be fatty acid converted over to carbohydrates and broken down. And the processes involve glycolysis, pyruvate oxidation, the Krebs cycle, also called the TCA cycle or carboxylic acid cycle, and then the electron transport chain. Now, if the availability of oxygen, if the tissue is so metabolically active that oxygen becomes a limiting resource, it's not enough of it is being delivered, and it's not enough is available, then the tissue, and that can happen in muscle, then the tissue starts going into what's called aerobic respiration, anaerobic respiration, the generation of ATP in the absence of oxygen. And in this case, it is no different, exactly the same as what is better known as, let's do it this way here. This, in fact, is the same as what is better known as lactic acid fermentation. Okay, lactic acid fermentation. It's important to realize that aerobic respiration is much more efficient way to produce ATP than lactic acid fermentation, and the production of ATP by this process, fermentation, is not particularly productive and cannot be sustained for very long. I put fermentation. It's not fermentation, it's fermentation. Okay. So the only actual source of the energy for contracting muscle is ATP. Those stores of ATP in the muscle, as I said earlier, get exhausted in roughly 5 seconds, 4 to 6 seconds. They can be regenerated. More ATP can be built up by substrate level phosphorylation of creatine phosphate of ATP by the donation of phosphate from creatinine phosphate, but that only lasts about 15 minutes in an actively active tissue. Then after that, and it's not one after another, but the aerobic respiration will be occurring right away, right from the start also. So aerobic respiration, which can be carried on for a long time if you're doing an aerobic kind of contraction of the muscle. And that can be sustained for many hours, as long as you don't exceed the ability of the muscle to generate ATP or accessibility of oxygen. You know, if you do, then the metabolic processes will start switching over to anaerobic respiration or anaerobic glycolysis, also called lactic acid fermentation. And this diagram shows that. It shows, of course, ATP can be immediately used, or ATP can be produced from the transfer of a phosphate group from creatinine phosphate to ADP, forming therefore ATP. But this can only last for about 15 minutes. Did I say minutes? Seconds. Let's get this a little darker so people see it. 15 seconds total reserves. So that's the first thing that gets used up. The next process that's going to be occurring simultaneous with this, right from the beginning, simultaneous with this one, would be aerobic respiration, or aerobic cellular respiration, in which glycolysis breaks down glucose to pyruvic acid. Pyruvic acid, that's not very good, is that? To pyruvic acid, and pyruvic acid is converted to acetyl-CoA, and then acetyl-CoA feeds into the Krebs cycle in the mitochondria, and you get followed by the electron transport chain, and you get generation of a significant amount of ATP in this way. You get about 36 ATP molecules per glucose molecule that gets metabolized or broken down. Now, and this is a process that is aerobic, it requires oxygen, and if this becomes limiting in any way, then you start going to, again, the breakdown of glucose by glycolysis, but this really is this exact same here and here. There's no difference, but then the pyruvic acid that gets generated does not go into the Krebs cycle. It gets fermented or processed into lactic acid, and this is a process that can produce ATP, but not very much. It's not terribly efficient, and you cannot carry that out to any large extent where you really have exhausted this mechanism of aerobic respiration and are counting completely on this one. You won't go very long like that. So first, using up ATP sources, At the same time, also generating some ATP from the dephosphorylation or transfer of phosphate from creatinine phosphate. At the same time, also generating ATP by aerobic respiration, this mechanism here, and quite a bit gets generated, but eventually exhausting the supplies of something, usually oxygen, could be glucose, I suppose, but usually oxygen, and then switching over as that becomes limiting to lactic acid fermentation, anaerobic respiration, which does not really at all last very long, 30 to 60 seconds, just lactic acid fermentation. Well, you can do an awful lot in 30 to 60 seconds, mind you, if it's a burst of energy that you need. Okay, let's move on. I have no idea. Sorry that I missed that one slide. I will go back later and check. I might have to make some comment during the Zoom meeting associated with the next Zoom meeting. Okay. One thing I do want to point out is that anaerobic glycolysis or anaerobic respiration will start kicking in when the muscle contractile activity reaches about 70% of maximum. At that point, the compression, bulging muscle, constant relaxation, contraction of muscles starts to compress blood vessels too much, and you don't get enough flow. You're not getting adequate flow necessarily if you're really compressing muscles continuously. Oxygen delivery gets impaired. You might be trying to generate too much force and you just can't maintain it because of the deficit of oxygen. And so pyruvic acid, instead of feeding into going towards acetyl-CoA production and feeding into the Krebs cycle, starts going to lactic acid fermentation. So you switch over, and it's a continuum. It's not like aerobic and then switched off and then anaerobic switched on. It's a shift over to anaerobic respiration or anaerobic glycolysis. Now, lactic acid that is produced can actually, in some ways, the body does respond in a positive sense because of, I don't know if you learned about this, but you hopefully did, about the, oh no, I talked to you about it in respiratory physiology and also blood, that you get the phenomenon of the Bohr shift, where the hemoglobin oxygen dissociation curve shifts to the lower right, so that in a lower pH environment, more oxygen can dissociate from hemoglobin when the blood gets into the tissues. If the pH is lower, more oxygen gets released, and this does occur in very actively metabolically active muscle tissue, because if it's really highly metabolically active, it'll start going to lactic acid fermentation, and lactic acid will drop the pH and you'll get that Bohr shift and more oxygen available. So it does sort of help a little bit. It does prolong the ability to carry out some more aerobic respiration, but in the end it does, you can't sustain it for very long and you end up stopping. So the muscle has to stop, it's fatigued, it has to stop contracting. The lactic acid will in fact diffuse out of the tissue into the bloodstream and get carried away, and this occurs mostly in the liver, but can occur also in the kidneys and heart. It gets converted back from lactic acid back into pyruvic acid in the liver. Okay, so we were talking really about muscle fatigue when we talked about that spectrum, kind of gradual shift over to lactic acid fermentation. And it's occurring, fatigue in fact is occurring because the ATP production cannot keep pace with the use of energy, the usage of the depletion of ATP. So you get into a deficit of ATP. You can't generate enough to generate muscle contraction, and you get an accumulation of lactic acid in the muscle. You cannot accumulate too much. Yes, you have that Bohr shift, which helps sustain things for a little bit, but you get a lactic acid accumulation, and you can't sustain it. So you get ionic imbalances because, after all, the sodium-potassium pump cannot reestablish the ionic concentration gradient required between the cells, the sodium potassium pump is operating on the basis of requiring energy, so that ATP deficit also causes ionic imbalances in the tissue, and the muscle starts to fatigue. It's slowing down in its ability to contract. So very intense exercise will cause a rapid onset of muscle fatigue, but it has typically a rapid recovery. As I said before, sodium potassium pumps are unable to keep up with trying to rebalance the ionic concentrations, and that's with very intense physical exertion. With low intensity exercise, you can get a much slower developing fatigue, and that's why people can run marathons. They can run for hours, literally, and yet still be able to, because it's a lesser intensity, they're not exceeding their ability to produce enough ATP. They're not exceeding their ability to deliver enough oxygen for aerobic respiration to occur. You get an oxygen debt if you exceed the ability to produce energy, if you exceed the ability of the muscle to pump for the sodium potassium pump activity. So you go into oxygen debt, and you don't have enough ATP. You stop whatever muscle contraction, skeletal muscle contraction that you were doing, running, whatever kind of exercise. And then you have to restore that. You have to bring yourself out of oxygen debt. The oxygen reserves have to be restored. So what you have to do, some of the oxygen myoglobin is used up. You have to restore that. The lactic acid that built up, and remember, pH is very important. It can change the activity of enzymes, so it can disrupt enzyme activity if the pH goes too low. So the lactic acid has to diffuse out of the muscle into the bloodstream and get carried to the liver, where it gets converted to pyruvate or pyruvic acid. Glycogen stores in the muscles have to be restored. The glycogen is and you should have learned this before, the glycogen is a polymer of glucose molecules all strung together into a very large polymer or polysaccharide molecule. It's a carbohydrate polymer, polysaccharide, and it's a source of glucose within the muscle. There's storage of glycogen in two parts of the body, in the liver and in the muscle. And in the two tissues, it has a different purpose. In the liver, it's there as a source of glucose that can be released into the bloodstream if your blood glucose levels drop below a certain amount, a minimum amount. If it gets too low, then glucose gets mobilized from glycogen stores in the liver. But in the muscle, it's there to provide carbohydrate for aerobic respiration and anaerobic or fermentation, lactic acid fermentation processes to occur. You have to also, following a generation of oxygen debt through active muscle and muscle going into muscle fatigue, you have to regenerate the ATP levels that are normally there and the creatine phosphate levels that are normally there. So oxygen debt, strictly speaking, refers to the extra amount of O2 needed for these kind of restorative processes so that you build up enough oxygen again and enough ATP again to overcome these problems that occurred with muscle fatigue.